So hello, my name is Adi, and today I'm going to talk about my project, which is improving the photosynthetic efficiency of non-model plants by editing the protein Rubisco. So first we need to ask ourselves why. Why do we want to go through this trouble of editing, of like improving photosynthesis? Well, the answer is deforestation. So deforestation is a huge problem that's had many negative effects. And one of these effects is by lowering the amount of carbon assimilated by trees and releasing carbon back into the atmosphere from trees that they cut down. And to contain climate change, it's necessary that we control the amount of CO2 that's in the atmosphere. And to do this, I decided that we should look at just the rate of photosynthesis in plants and just like try to improve that. And photosynthesis is the reaction where carbon dioxide is taken in by plants and oxygen is released. So I wanted to edit that to make it faster. So yeah, for our goal for this project was to find an effective method to engineer Rubisco, which is often the rate limiting step in photosynthesis. Rubisco is the protein that takes in carbon dioxide or oxygen and starts the reaction. When Rubisco takes in carbon dioxide, it undergoes photosynthesis and goes through the Calvin cycle, which is what we want. It releases oxygen. But when it takes in oxygen, it goes through something that you and me go through as well. It's called cellular respiration, and then you release carbon dioxide. We don't want we don't want plants to do that. So I wanted to engineer Rubisco to become more specific, to select to just take in more carbon dioxide, and to also just become faster, because oftentimes Rubisco is a pretty slow protein. So to do this, we looked at papers that use directed evolution, protein engineering, genetic engineering, and other methods. And through this, we wanted to increase the carbon assimilation rate of plants and also improve the carbon sequestration of forests, which is basically how much carbon is stored within forests. And through this, we lessen the effect of climate change. So these are the methods explored. We looked at directed evolution, mostly through using E. coli as screens. We looked at protein engineering projects which changed the protein's shape and changed the charges of different amino acids just to change the shape and just change the reaction in general. And we also looked at experiments which transplanted different Rubisco types from more efficient plants into less efficient plants. And there were also a few other papers that changed metabolic pathways and other things. Okay, so directed evolution and protein engineering. So there were many targets for this, mostly from prokaryotes and cyanobacteria, because Rubisco from these organisms tend to be simpler and just easier to engineer. So these were TE Rubisco, SE Rubisco, RS Rubisco, and Rubisco from M. bertani. And basically for these, we used RDE screens, which are basically Rubisco-dependent E. coli screens, where the E. coli don't have Rubisco to provide, to start the Calvin cycle and provide energy. So they need non-native Rubisco, and these are used in labs. So basically the way they work is that they use PRK to increase the toxicity inside the cell, and basically the reaction that's started by Rubisco will lower the toxicity by activating RuBP. And basically through this, we'd select for more and more efficient forms of Rubisco, which were either more selective or faster or something, or sometimes both. So here we found that there were around 3 different screens that were used. There was RDE1, which is the first one, which had a false positive rate of 0.5%, because oftentimes there would be mutations which would cause there to be like the PRK gene would just be deactivated, and then there would be no checking for toxicity. Then there was an RDE MM1 screen, which used a GAP-A gene to prevent that, to prevent a flux like that, and to just like, it lowered the false positive rate to 0.1%. And these two were often used for TE Rubisco and SE Rubisco because the environments seem to favor these two Rubiscos more. RDE3 is basically an evolved form of RDE2, which uses fusion of PRK and MPT2, which are two different toxins, to select for it. And M. Bartani used the RDE MM1 screen. And basically, the strains from these various experiments tended to be less viable from the wild type, as you can see here. Although that they might have been more specific sometimes or faster, there was often like a huge trade-off. Like if it was faster, they might lose like a lot of their selection ability, and they would just start selecting for oxygen, which we don't want. Um, and as you can see here, the plants that were edited, this is the Embertani one, were a lot smaller than the wild type in general. So here we can see a comparison. And basically, even sometimes if they did work, they need very specific conditions, like they would need a lab setting with a really high concentration of CO2, which just doesn't occur in the real world. So we eventually looked away from dark red evolution because it just didn't seem like it was completely feasible as it was right now. Oh, okay. So then we looked at using Rubisco from other species, which might be more efficient and already evolved for just like better photosynthesis. So for this, we found that red algae is often used because Rubisco from red algae is often a lot more selective, almost 2 times more selective in some cases, and is also a lot faster than plant Rubisco. So to do this, we carried the genes from the red algae and knocked them in place of the RBCL gene in Rubisco in the plants. And basically, the RBCL gene codes for the large subunit of Rubisco. Rubisco is made of large subunits and small subunits. And we knocked out the large one because when we knock out the large one, typically it's less error prone. However, when we looked at this, when we knocked in the large subunit of the red algae, they just wouldn't form a proper tertiary structure because there was a lack of suitable binding enzymes, and they just wouldn't fold properly. And we also looked at then we looked at just taking in the entire genome, not just the large subunit, and that seemed to work, as you can see here. However, it was just less dense. There was like a lower concentration of Rubisco in these plants, and it just seemed less fit overall. So although this seemed to work, it just didn't seem to get like there was no concentration that was higher than this that was found. So it was just less fit than the wild type in general. So it didn't seem to work. There was also an experiment which used sunflower, and that it did work. It was a faster photosynthesis rate. However, the plants grew to be about 24% smaller than the wild type. So that was also less fit. So after looking at various experiments, it did seem that although Rubisco has been a focus for decades, it doesn't seem to be a completely viable focus for the future, as there is a difficulty in modifying the enzyme properly to fit within all the processes of the cells. And other routes have been shown to be more effective, such as editing the metabolic pathways apart from the Calvin-Benson cycle, which is what we looked at here. A C3 to C2 conversion, which are different forms of photosynthesis, may be more viable in the future, as proteins such as BHAC seem to be able to create a reaction similar to C4 photosynthesis. And also editing pathways such as ECGC pathways increase biomass by about 30 to 50% in rice. So these all seem to be more promising than editing Rubisco. Thank you for listening. And special thanks to Henry.
Thank you, Adi. Couple questions from the audience, but I'm going to hold on those just to keep us on time because I want to make sure that we wrap up so that everyone can head over to our keynote when it starts. But thank you very much. I'm going to send the questions to you after the fact, if that's okay.